Absorption of any substance in the nutrition process involves two phases. So, food is coming through the lumen of the gastrointestinal tract. So, this is a part of the intestine which is shown, and this region is called as the lumen through which the food, digested food, will be coming. So, first step is from this lumen the nutrients are to be absorbed into these epithelial cells. So, they are to be first absorbed into the epithelial cells of the villi. So, that is the first phase of absorption. Now, the second phase. So, this is an enlarged structure of a villus. Inside the villus, we can see the capillary network as well as the lymph capillary, which is called as the lacteal. During the second phase, so the first phase is now over. From the lumen, the nutrients have come into the cytoplasm of these intestinal epithelial cells. So, here these are the intestinal epithelial cells. From there, it has to go to the blood capillary, or it has to go to the circulation, so that it is transported to all parts of the body. For this, there is a blood capillary network as well as there is a lymphatic vessel that is running through the center of the villus. So, the second phase is from the cytoplasm of these intestinal epithelial cells, the nutrients have to enter either into the bloodstream or into the lymph vessel. So, that is the second phase of absorption. So, first is from lumen to the epithelial cells, that is from here to the intestinal epithelial cellular cytoplasm. Second is from these cells, that is which is shown here, they are to be taken up either by the blood capillary or the lacteal. Digestion is one of the major functions of gastrointestinal processes or nutrition, because it is the breakdown of large complex macromolecules in our food to simple substances. So, why they are converted to simple substances? Because the nutrients in the food which we ingest cannot be as such absorbed. So, they are to be converted to the absorbable form, and once they are to be in the absorbable form, we need to break them down. That process is called as digestion, which is mediated with the help of digestive enzymes. And there comes the next part, which is called as absorption. So, we will study how the absorption of end products of carbohydrate, fat and protein digestion takes place in our gastrointestinal tract. So, we first go to the absorption of carbohydrates. All the carbohydrates in the food are first broken down to monosaccharides, and the food is absorbable, or the carbohydrates are absorbable only if they are converted to monosaccharides, and very small fraction of disaccharides may be absorbed, but majority of absorption will take place only if they are in monosaccharide form. And which are the main monosaccharides which we get as end products of carbohydrate digestion? They are glucose, fructose and galactose. Glucose is obtained from the digestion of starch, maltose, lactose. Lactose is milk sugar, sucrose, etc. Even by the digestion of sucrose, we are getting glucose. Whereas we get galactose from the digestion of lactose. Then fructose is obtained by the digestion of sucrose or cane sugar. So, finally, these three are the monosaccharides which are derived from carbohydrate digestion. They are glucose, fructose and galactose. So, 80% is glucose, 20% of monosaccharide absorption accounts for that of galactose and fructose. 80% of absorption takes place for glucose. It is because glucose is the final digestion product of most abundant carbohydrate food, that is the starch, and its digestion releases glucose, and so 80% of absorbed monosaccharides is in the form of glucose. Now we go to how absorption takes place. Glucose is absorbed mostly by secondary active transport, which is also called as co-transport, that is, it is associated with the transport of sodium. There is a protein called as the sodium glucose symporter, which will transport sodium along with glucose. So, that is called as symport or co-transport. Galactose is also absorbed through the same mechanism by another protein which is called as the sodium galactose importer. The mechanism operational is the same, but the proteins will differ, the transporters will differ. Whereas fructose is not absorbed by co-transport. It is absorbed through facilitated diffusion. Most of the fructose is absorbed through facilitated diffusion with the help of a permease located in the cell membrane of the intestinal epithelial cells, and this is an energy independent process because it is facilitated diffusion. So here there is no coupling of fructose transport with sodium transport. And what happens to this fructose once it is absorbed into the intestinal epithelial cell? Inside the intestinal epithelial cell, the fructose will first be phosphorylated into fructose phosphate, and then it is converted to glucose phosphate, and then finally to glucose. That means there is conversion of fructose into glucose, and it is absorbed through blood in the form of glucose. So even if it is absorbed into the intestinal epithelium as fructose, it is converted inside the intestinal epithelial cells into glucose and then taken up by the blood. And fructose is not co-transported with sodium, it is transported only through facilitated diffusion, and so the rate of absorption of fructose is about half that of glucose or galactose. We will see how the absorption of glucose takes place through sodium glucose symporter. So for that, we have to first go to this particular protein located on the cell membrane of the intestinal epithelial cells, and this is called as the sodium potassium pump, which is an active transporter. It's a pump which works on the hydrolysis of ATP, and the energy derived from the hydrolysis of ATP is used for its working, and it will pump 3 sodium ions out of the cell and take in 2 K plus ions into the cell. So the transport of sodium as well as potassium is against the concentration gradient, that is why it is using energy. So this is forcibly pumping 3 sodium out to the cell exterior. So the cell exterior will be having a higher concentration of sodium ions. So this causes a tendency for sodium to come in. So the sodium will have a diffusion energy to come in. So this diffusion energy which sodium has to come in will be utilized in another way. That is, here you can see there is active transport of sodium with the help of the sodium potassium pump into the spaces between the cells and to the lumen. Because of all these, the sodium will have a tendency to come into the cell interior, and for that the sodium will go for another protein located in the cell membrane, which is called as a sodium glucose symporter. So this symporter or this transport protein is having binding sites for sodium, but it will start working only if another molecule comes and bind to it, that is glucose molecule. So this sodium glucose symporter will be operational only if sodium and glucose binds to it. So once both these molecules are bound to this symporter, the symporter will undergo some conformational changes, and thereby it brings in sodium as well as glucose into the cell. So that is how glucose is transported. So here in the whole process ATP is utilized, because driven by the energy obtained by the hydrolysis of ATP, the sodium potassium pump has pumped out sodium ions to the cell exterior, and therefore the sodium ions had a tendency to come into the cell interior. For that, the sodium ion will come and bind to the sodium glucose symporter, and the symporter starts working when glucose binds to it, and when it starts working, both sodium and glucose will be transported into the cell. So this is called as symport or co-transport. This is energy dependent, but it is not this transporter which is utilizing energy. It is utilizing the diffusion energy created by this pump which worked on ATP. So we can say the entire process is dependent upon ATP, but the symporter is not directly ATP dependent. So that is what is called as the sodium glucose symport, and similar way of symport is resorted to by galactose also for its transport. So here we can see that the sodium ion will combine with the symporter, which will not transport sodium into the cell interior until it combines with glucose, and once glucose comes and binds, the sodium and glucose molecules are transported together into the cell interior, and the low concentration of sodium inside the cell actually dragged sodium to the cell interior along with glucose. And once the glucose is inside the epithelial cell, now it has now come only into the epithelial cell. Now it has to go to blood. So in the epithelial cell membrane, there will be some transport proteins and also some enzymes. They will help in facilitated diffusion of this glucose into the spaces between the cells, and from there it will get into the blood. So the transport proteins and enzymes present in the intestinal epithelial cells will now take this glucose to the space in between them, and from there blood will take up glucose. So there are two phases of glucose transport from lumen to the blood. That is, first from the lumen into the intestinal epithelial cell through symport, and from the intestinal epithelial cell to blood through facilitated diffusion. So these are the basolateral spaces, that is the lateral sides of the intestinal epithelial cells will have spaces, interstitial spaces in between. These spaces are called as the basolateral spaces. Next is the absorption of proteins. Amino acids are the end products of protein digestion, but together with that there will be some dipeptides and tripeptides which remain after protein digestion, and mostly the proteins are absorbed in the form of amino acids, and rarely some in the form of dipeptides and tripeptides. And we first go to amino acid absorption. Most of the amino acids follow sodium co-transport mechanism, just like glucose was absorbed. So here there will be a sodium concentration gradient created by an ATP driven sodium potassium pump. This will give a diffusion energy for sodium, which will try to come to the cell interior, and for this it will resort to a transport molecule which is called as the sodium symporter, and this may be a sodium amino acid symporter instead of a sodium glucose importer, and that symporter will work only if sodium and the concerned amino acid comes and binds to it. So the mechanism is same as that of glucose absorption, but the transporter protein will vary, and these transporter proteins for sodium amino acid symport are at least of 5 types. A particular type of transporter will be specialized for taking up acidic amino acids like dry carboxylic acids like aspartic acid, glutamic acid, etc. There is another type of sodium amino acid symporter which is specialized to take up basic amino acids like cysteine, lysine, arginine, etc. The third one is specialized to take up neutral amino acids along with sodium. The neutral amino acids such as alanine, valine, serine, isoleucine, etc. And there is a fourth one which is specialized to take up sodium and amino acids like proline. And the fifth one is specialized to take up peptides, that is the dipeptides or tripeptides. So most of peptide absorption as well as amino acid absorption depends upon sodium amino acid or sodium peptide symporter system. Whereas there are a few amino acids which do not require the sodium co-transport mechanism, and they are transported through special permeases or membrane proteins or carrier proteins which are located in the cell membrane by facilitated diffusion. So we can summarize it in a nutshell that majority of the amino acids and peptides are taken up by sodium co-transport, whereas a few are taken up by facilitated diffusion. So just as it was mentioned earlier in the case of glucose, these transporters bind to amino acids only after they bind with sodium, and once it is fully loaded with sodium and amino acid, it will undergo a conformational change that will pump sodium and amino acid, that will dump sodium and amino acid into the cytoplasm, and it will reorient back to its normal configuration, and the process is repeated. So here also the absorption of amino acids is dependent upon the electrochemical gradient of sodium, which was initially created by the sodium potassium pump. So this is also an example for secondary active transport. And the intestinal epithelial cell, its basal side as well as the lateral side, the basolateral membrane of the intestinal epithelium contain other transporters which will help in transporting the amino acids further from the cell cytoplasm into the blood. Because as it was mentioned in the case of glucose and other monosaccharides, here also there are two phases of absorption, that is one from the lumen to the cell and second from the cell to blood. So, for the second phase of transport there will be additional transporters which may allow facilitated diffusion or free symbol diffusion into blood, from the cell into the blood. So, that is how the amino acids are absorbed. And this symporting mechanism is operational in the case of L amino acids, but D amino acids do not depend upon carrier mediated active transport. They are absorbed by passive diffusion. The naturally occurring L or levorotatory amino acids are absorbed by carrier mediated active transport. And yet another mechanism, yet another important point to be mentioned is that the absorption of peptides. Usually peptides which are longer than 4 amino acids are not absorbed. They have to be converted into amino acids, either dipeptides or tripeptides or amino acids, then only they will be taken up. So, peptides longer than 4 amino acids are not usually absorbed, and certain peptides show a particular behavior, that is, there are some peptides which are absorbed more easily in their peptide form rather than getting them converted to amino acids. An example is a peptide which is called as carnosin. Carnosin is a dipeptide, and this carnosin has got two monomers. One is called as beta alanine and the other is called as L-histidine. And it has been revealed that the absorption of carnosin, that is the dipeptide, is faster than the independent absorption of either beta alanine or L-histidine, which are its monomeric amino acids. So, this is an example where a dipeptide is absorbed faster than its monomeric amino acids. So, certain peptides show such behavior, that is dipeptides of alanine, dipeptides of histidine, dipeptides of methionine, dipeptides of glycine, etc. are absorbed more easily in the dipeptide form rather than as free amino acids, and carnosin is the best example as it was cited earlier. And these dipeptides and tripeptides are also taken up by co-transport, and sometimes they are taken up along with H plus ions rather than sodium ions. So, that will be H plus dipeptide co-transport, and the transporter which operates in that case is called as pepT1. Otherwise, they will follow sodium in the sodium peptide co-transporter mechanism, and sometimes they are also taken up along with H plus ions through its importer which is called as pepT1. And once, so the first phase is over, that is from the lumen they are taken into the intestinal epithelial cell. Once they come inside the intestinal epithelial cell, these absorbed dipeptides and tripeptides are digested into amino acids. By whom? There will be enzymes inside the cytoplasm of the intestinal epithelial cells. So, the enzymes present in the intestinal epithelial cell cytoplasm will further break down the dipeptides and tripeptides into amino acids, and from there the amino acids are taken up to the blood. So, the dipeptides and tripeptides can be absorbed from the lumen into the intestinal epithelial cell, but they cannot be absorbed into the blood. For getting absorbed into the blood, they are to be first broken down to amino acids. Now, coming to the absorption of proteins as such. The absorption of proteins as such is a very rare mechanism, but we can see this phenomenon in newborn babies. For a few days after their birth, the newborn babies, they are showing ability to absorb proteins as such. This is an adaptive feature because in the first few days of lactation, they are given the mother's milk, which is a yellow colored fluid which is called colostrum, and the colostrum is rich in antibodies, and the antibodies are proteins. They are antibodies are glycoproteins, and so these antibodies can, without digestion, can be as such absorbed into the infant's digestive tract. So this is an adaptation. So this will allow the newborn baby to acquire immunity because it is getting antibodies from the mother's milk, mother's first milk or colostrum, and it is as such absorbed without digestion. But later this property is lost by the small intestine, that is the ability to absorb the protein in intact form. That ability is later lost and that process is called as closure. So in the initial days it is very essential that, or it is recommended that the baby is to be given colostrum, because it is only that way through which it is getting antibodies in their own intact form. And later this property is lost and the antibody will be digested into amino acids and then only it will be absorbed. But if it is given this colostrum during the first few days of lactation, it is ensured that all these antibodies in the colostrum are as such absorbed. So babies who do not receive colostrum are having a higher risk of getting some opportunistic infections. So this is the advantage of taking up proteins as such in newborn babies. And the newborn baby, the digestive system or the small intestine of the newborn baby is also able to take up some egg albumin for a small period after their birth. So these are the various techniques by which proteins are taken up. Another point to be noted in the absorption of amino acids is that most of the amino acids, almost all the amino acids are taken up by the intestinal epithelium without any chemical change. But there are two exceptions. They are glutamic acid and aspartic acid. So before getting absorbed, glutamic acid and aspartic acid undergo a transamination. Transamination is a process by which the amino group of aspartic acid and glutamic acid are donated to pyruvic acid and thereby releasing alanine. So alanine is released and it is taken up in that form. And suppose there is low rate of absorption of these substances, that is glutamic acid and aspartic acid, then the mechanism is adjusted in such a way that if the rate of absorption is low, the rate of transamination will increase so that more and more absorption is facilitated. So we can say that the rate of transamination of glutamic acid and aspartic acid is inversely proportional to the rate of absorption. If the absorption rate is low, then the transamination would be fast and that will compensate for the low rate of absorption. Next is the absorption of fats. So before that we will go to the end products of fat digestion. That is, most of the neutral fats in our diet are in the form of triglycerides, and when they are digested, the end products are, if they are fully hydrolyzed or fully digested, we will be getting glycerol and 3 fatty acids. Or if they are partly hydrolyzed, we will be getting diglycerides and monoglycerides. So we can summarize that, or we can say that the final end products of fat digestion will be glycerol, fatty acids, diglycerides, monoglycerides. Out of these, glycerol is easily soluble, so its absorption doesn't cause any problem. Short chain fatty acids, their absorption will also take place by simple diffusion through the epithelial cell membrane. The problem is encountered by the long chain fatty acids. So we will go to how these substances are getting absorbed. So the main problem is their water insolubility. So they are to be made water soluble first. So before that, we go to one more point, that is when the triglycerides are hydrolyzed, we get the end products which I just mentioned earlier, that is glycerol, monoglycerides, diglycerides, fatty acids, etc. But the lipases which carry out this hydrolysis process are reversible in nature. So when digestion takes place, these end products will be formed, and if the end products are not removed from that place, the process can reverse again. They can be reconstituted into triglycerides. So this has to be avoided, or this problem is to be overcome. That problem is overcome by the bile salts, because the bile salts are already there in action for the process which is called as emulsification. What is emulsification? It is the breakdown of large droplets of fat into simple substances, into smaller substances. That is what is called as emulsification. If only the fats are emulsified, the lipids will be able to act effectively on them. So that process is being carried out there, and the bile salts are there already in action for emulsification. And there comes the next problem, that is after digestion of triglycerides, you get diglycerides, monoglycerides, fatty acids, glycerol, etc. But since the enzyme is reversible, they can again be converted back to triglycerides. So these end products are to be removed from there. So for this process also, the bile salts come into help. That is, these bile salts, when there is enough concentration of water there, they will start forming spherical water soluble droplets which are called as micelles. So the bile salts will form water soluble spherical cylindrical globules which are about 3 to 6 nanometer in diameter, and these substances are called as micelles, and each micelle will be having at least 20 to 40 molecules of bile salt. And what are bile salts? Sodium glycocholate and sodium taurocholate are the bile salts present in bile. And now the micelles are formed. So what is the morphology of a micelle? The micelle will be having a central nucleus portion which is lipid soluble, or it is composed of sterols, and it is in this place that it stores the end products of fat digestion. And it will be having an outer polar region that is water soluble. So this micelle is a droplet which is having an outer water soluble portion and an inner core which is lipid soluble, and it is in this lipid soluble portion that is it has incorporated the end products of fat digestion. Now the whole droplet on its outside is water soluble. So the sterol nucleus is containing the fat digestate, that means the end products of fat digestion, and the polar groups of bile salts are projecting outward to cover the micelle. Now these polar groups are negatively charged, so they will have a tendency to dissolve in water, and there will be water already in the lumen which is derived from the digestive fluids, that is the digestive fluids like the intestinal juice, gastric juice, pancreatic juice, etc. will be having water. So these micelles will dissolve in that water and they remain stable there until it is the time for fat absorption. Now once digestion is over, these micelles, they will start acting like a ferry or a transport system. Okay, so what do they do? They have got all these end products hidden inside them in their sterol nucleus, and they have got an outer water soluble portion. So they start moving like a ferry through this water, and they will move towards the brush border of the intestinal epithelial cell. What is brush border? The microvilli present in the intestinal epithelial cells. So now they are acting like a boat, because there was one place where digestion was taking place. From there they got the digestion products. These products are taken into their nucleus, and now they are acting like a boat which will travel up to the microvilli of the intestinal epithelial cells, and the microvilli are fastly moving. So here what will this boat do? What is the actual service a boat does? A boat will carry passengers from one side of a river to the other side, and once the passengers they come down, or once they are alighted, the boat will come back or the boat will go back. Similarly do the bile salts behave. That is, once they come to the microvilli, these monoglycerides, fatty acids, etc., which are stored inside their sterol nucleus will come out. They will diffuse immediately out of the micelles, and they will go to the interior of the epithelial cells, and they will easily cross the membrane of the epithelial cells because they are lipid soluble and the membrane is lipid bilayer, and so they will easily enter into the intestinal epithelial cells. So the passengers are now unloaded, and now the ferry, that is the bile salts, will go back. So the bile salts are released back into the chyme, and they are used again and again for this ferrying process. This is exactly what a ferry or a boat service does. Okay, and so this process, the involvement of bile salts is very important in fat absorption. It is because of the presence of bile micelles that about 97% of the fat will get absorbed from our diet. If the bile salts were absent, if micelle formation was absent, only 40 to 50% will be absorbed. So here two phases of fat absorption has been covered. That is, there was a lumen phase in which the micelles were formed, and then there was a penetration phase during which the end products of fat digestion crossed the membrane of the intestinal epithelial cells and reached the cytoplasm of the intestinal epithelium. Now there is a third phase. That is, from there they have to be taken up into the circulatory system. So that can be called as the intracellular phase. So we will go to that. So as it was already mentioned, the bile salts, they play the same role as a ferry, and they also help in ferrying the end products of cholesterol digestion. That is, cholesterol is in the form of an ester. So that will be digested by the enzyme cholesterol esterase, and this cholesterol esterase will get hydrolyzed into cholesterol and fatty acids, and free cholesterol and fatty acids will be released in the small intestine. And now the phospholipids. The phospholipids are also esterified, and they will also be digested to form free fatty acids and phosphate groups. So for their transport also the bile salts come into help. So just like monoglycerides, triglycerides, fatty acids, etc., long chain fatty acids, etc., transported by the micelles, the end products of cholesterol and phospholipid digestion are also ferried towards the intestinal epithelial cells. Now all these substances have now reached the intestinal epithelial cells. Now they have to go to the next phase, that is, they are to be absorbed into the circulatory system. But to be taken up by the blood, they are to be first made water soluble, because these substances are lipid soluble. They can easily cross the membrane and come inside, but they cannot enter into the blood because they are water insoluble. So first they are to be made water soluble. So for this, the second phase of fat absorption takes place, that is, these substances, that is the fatty acids, monoglycerides, the diglycerides, cholesterol, the digested phospholipids, etc., will be taken to the smooth endoplasmic reticulum inside the cell. And inside the smooth endoplasmic reticulum, these substances will be rejoined into triglycerides. So they are again reconstructed into triglycerides, and they are integrated with cholesterol, phospholipids, and fat soluble vitamins also. So all these water insoluble substances are joined together. That is, they are reconstituted to triglycerides and integrated with cholesterol and phospholipid digestates, and then they are coated with proteins. Now they are droplets which are now water soluble. Earlier these individual substances were not water soluble, but now they have been reconstituted. They have been mixed with the cholesterol and phospholipid digestates. They have also been mixed with the fat soluble vitamins, and now they have been given protein coats. So they again form new droplets, which you can call as chylomicrons, which are water soluble. But now the problem is that even though the chylomicrons are water soluble, they cannot be taken up into the blood capillary. So here you can see the villus. This is the villus, and inside the villus you can see the capillary network, and you can see a central lymph vessel which is called as the lacteal. So this is the capillary network, and we have seen a central lymph vessel which is called as the lacteal. So all this process which I mentioned, that is the reconstitution of triglycerides, takes place in the intestinal epithelial cells. So from there it has to come to the blood. But the problem now is that once the chylomicrons are constructed, they are water soluble, but they are of bigger size. So they cannot be taken up by the blood capillary. Even if they are water soluble, they are too large to be taken up by the blood capillary. So what they do? They will be taken up by the central lymph vessel, which is called as lacteal. So that is the most important point to be noted, that is the majority of end products of fat digestion are not taken up by the blood capillary, but they are taken up by the central lymph vessel, which is called as lacteal. So they are directly absorbed to the lymph rather than by the blood. The lymph will later travel through the various lymph ducts present in the body, and finally it will drain into the blood. So it will come back to the blood circulatory system itself. It will come back to the circulating blood, but during the initial phase of absorption it is taken up by the lymph. So we can say chylomicrons are formed in the intracellular phase. So it is very important that we understand the difference between micelles and chylomicrons. Micelles are formed in the lumen of the intestine, whereas chylomicrons are formed inside the epithelial cells lining the intestine. So the micelles are formed out, but once they come in, once the end products of fat digestion come into the cell, there occurs the formation of chylomicrons. Both are water soluble. Chylomicrons are formed in the intracellular phase, whereas micelles are formed in the lumen phase. So these are the two droplets or two specialized droplets which are formed during fat absorption. That is the micelles formed by the bile salts and the chylomicrons formed inside the intestinal epithelial cells. Now, there are some fatty acids which need not be reconstituted or which need not be incorporated with the chylomicrons. So instead of getting incorporated with the chylomicrons, they will directly be absorbed into the blood. So they are not going directly to the lymph, but they are going to the blood. Some small quantities of short and medium chain fatty acids will be absorbed this way directly into the blood rather than into the lymph vessel. So the examples are the short and medium chain fatty acids which are derived from butter, fat, etc. are absorbed directly into the blood rather than into the lymph vessel. This is because the short chain fatty acids are water soluble, so they can directly get into the blood. They need not go for the reconstitution or chylomicron building process. Others will go for the process which is mentioned earlier. And the short chain fatty acids are directly absorbed into the intestinal epithelial cells and then to the blood capillaries. Next is the absorption of vitamins. The vitamins are not digested, but they are absorbed as such in the ingested form itself, and the absorption of vitamins occurs in the anterior part of the small intestine. And there are two types of vitamins. They are fat soluble and water soluble vitamins who are getting absorbed by different mechanisms. So first we go to the fat soluble vitamins. They will dissolve in the micelles just as the end products of fat digestion. So they also dissolve into the central sterol nucleus of the micelles, and they are absorbed passively along with the end products of fat digestion through the intestinal epithelial cells. So the first phase is over. Now from the intestinal epithelial cells, so from the lumen now they have come to the intestinal epithelial cells, and from the intestinal epithelial cells they have to go to the next phase, that is to enter into the circulatory system. So that is the absorption regarding the fat soluble vitamins. Now the water soluble vitamins, they are mostly absorbed by passive transport, and here one which is worth mentioning is the absorption of vitamin B12, which is also called as cyanocobalamin. It is a cobalt containing vitamin, and there are two ways of its absorption. A small portion of vitamin B12 in the diet will be absorbed by bulk transport, that is by pinocytosis or cell drinking, since it is water soluble. Majority of its absorption is carrier mediated, and that carrier which helps in absorption of vitamin B12 is called as Cassell's intrinsic factor. It is a protein, it is a glycoprotein which is secreted by the stomach. In the stomach there are gastric glands, and in the gastric glands there are three types of cells. The chief cells produce pepsinogen, that is the protein digesting enzyme. The parietal cells produce HCl, and there are goblet cells which produce mucus. So there are three types of cells in the gastric glands the chief cells producing pepsinogen, the parietal cells producing HCl, and the goblet cells producing mucus. These parietal cells which produce HCl also produce a protein which is called as Cassell's intrinsic factor, and it is this Cassell's intrinsic factor that helps in absorption of vitamin B12. Now we will go to the various steps in the absorption of vitamin B12. So this is the vitamin B12 that is coming through the esophagus to the stomach. So this is the dietary vitamin B12. Here we can see that it is now complexing with a protein. This protein is called as R binder. So this shaded protein which we can see here is called as R binder or transcobalamin 1, which is synthesized by the salivary glands as well as the gastric mucosa. So this is the R binder which is produced by the salivary gland. This is the R binder which is produced by the gastric mucosa. So it is to this protein that cyanocobalamin or vitamin B12 which gets first complexed with. Now why is this complex formed? Vitamin B12 can enter into duodenum only by binding with this R binder. So R binder helps the transport of vitamin B12 from the stomach to the small intestine. Now when this complex passes through the stomach, the parietal cells of the gastric glands of the stomach also produce the Cassell's intrinsic factor. So there is already R binder which is produced by the salivary gland, which is also produced by the gastric mucosa, and it is to this R binder that vitamin B12 is bound to. Only in this bound form it can enter into the duodenum. Now when all these things pass through the stomach on its way to the duodenum, the stomach is also producing the Cassell's intrinsic factor. It will also move to the duodenum along with this complex. Now when the B12R binder complex reaches the duodenum, the pancreas produces certain proteases which will hydrolyze or digest the R binder protein. The vitamin B12 is now free. So this B12 required this R binder only as a transporter to come to this duodenum. Now it is no more useful. So this R binder will be digested by proteases which are produced by the pancreas. Now the B12 is free, but it cannot be absorbed as such. B12 cannot be taken up by the small intestine if it is in free form. Now this B12 will bind to the Cassell's intrinsic factor which has been produced by the parietal cells of the stomach, and the Cassell's intrinsic factor had also moved along with this complex to the duodenum. So there they meet. Even if they are meeting here, they are not complexing. But once they reach the duodenum and once the R binder goes away by hydrolysis, this Cassell's intrinsic factor will bind with vitamin B12. This forms a complex. Now this complex is taken up by the small intestine. For this, the small intestine contains certain receptor proteins. So the receptor proteins present on the ileum, the remaining part or this ileal part, there will be receptor proteins on the membrane or the epithelium of this ileum, and it is to this receptor protein that this vitamin B12 intrinsic factor has to bind to, and from there it is absorbed. So if B12 is not bound to the intrinsic factor, it cannot be absorbed, because here you can see that the membrane of the ileum is directly interacting with the intrinsic factor rather than B12. Okay, so intrinsic factor is required, or it is essential for B12 absorption, otherwise it won't be absorbed, because here there is a receptor to which only the intrinsic factor can bind. So without the involvement of intrinsic factor, vitamin B12 will not be able to enter to the intestinal epithelial cells. So it is here that the absorption of B12 takes place. So this taking up of vitamin B12 intrinsic factor complex by the cells present on the wall of the small intestine, for taking up this complex, there are certain factors required. There should be an optimum pH, there should be sufficient concentration of calcium and magnesium ions. So, the taking up of vitamin B12 intrinsic factor by the small intestine, by the ileum, is dependent upon optimum pH and optimum concentration of calcium and magnesium. So, from this it is clear that it is the Cassell's intrinsic factor that is quintessential for the, or it is essential for the absorption of vitamin B12 into the intestine. Suppose there is non-secretion of Cassell's intrinsic factor, it will lead to malabsorption of vitamin B12, which will lead to deficiency of vitamin B12, which can lead to hematopoietic disorders, that means blood related disorders. Example is pernicious anemia, that means the formation of RBCs will not be completed and most of our RBCs will be immature. That condition is called as pernicious anemia. And deficiency of vitamin B12 can also lead to spinal cord related neuropathies. So, these are the various mechanisms by which the different types of vitamins are absorbed. Now, we move on to, so all this is about absorption in the small intestine. That is, we saw how water is taken up, ions are taken up, carbohydrate end products are taken up, the protein end products are taken up, the end products of fat digestion are taken up, and how the vitamins are absorbed. They all take place in the small intestine. Now, we will go to the phase of absorption which occurs in the large intestine. So, the fluid that comes out of the small intestine and that which enters the large intestine is called as chyle. So, it is almost fully digested and majority of nutrients have been absorbed, and it is the chyle that reaches the large intestine. So, once it reaches the colon of the large intestine, most of the water and electrolytes will be absorbed in the colon also. So, colon is specialized for the absorption of water and electrolytes, and thereby it will leave less than 100 ml of fluid to go with the feces, and the rest will be absorbed by the colon. So, majority of water absorption also takes place here. Ions are also absorbed here, and it may leave only 1 to 5 milliequivalents of sodium and chloride ions to be lost in feces. The rest will be absorbed in the colon itself. So, we can say that most of the absorption in the large intestine occurs in the initial or the proximal part of the large intestine, whereas in the distal part of the large intestine or colon, the feces starts getting formed and stored.